So yes, my main focus, because it's my main research area, has been for most of my career, is the impact of neuropeptides, both in animal models, but now pretty much exclusively in humans on neural networks. And today I'm going to share with you some new data where we directly actually look at corresponding changes from a task and also a resting state. Oops. Okay. Neuropeptides are interesting in that they have no reuptake mechanisms, and therefore they're found in pretty high concentrations in the CSF. That means that they can actually also coordinate activity both within regions, because they can travel quite a long way without the reuptake mechanisms, and they're very important in terms of presynaptic modulation of classical transmitters. And also they can have paracrine actions via the cerebroventricular system itself, which can theoretically allow them to alter global brain states. Unfortunately, there hasn't been a lot of work looking directly at neuropeptide effects. They're almost all given by intranasal infusion in humans. And I just wanted to summarize them very quickly for you and then focus on oxytocin, which is the only peptide where we have really sufficient information to provide some realistic comparisons. But insulin's been looked at, for example, resting state, very interesting increase in functional connectivity between medial prefrontal and hippocampus, which is associated with improved cognitive function. Vasopressin so far hasn't been looked at in resting state, and it has rather inconsistent effects on changes in amygdala and insula, so this is still much an open area with tasks. But the one I'm going to focus on is oxytocin, and although many effects have been reported, I want to focus particularly on the main ones, the most reproducible ones. And in terms of resting state, this is an enhanced or increased functional connectivity between parts of the medial prefrontal cortex and the amygdala. And in task, at least particularly in males, the amygdala shows a decreased activation in response to negative faces and scenes, particularly fear ones. Why is this important? Well, we already know from anxiety and depression disorders that enhanced amygdala responses to negative emotional faces and scenes is very much something that occurs regularly, and we want to try and prevent it. And also there is considerable evidence for reduced top-down emotional control, which is reflected in weakened functional connectivity between prefrontal regions and amygdala. And Ben in his talk was obviously talking about using real-time fMRI with neurofeedback as a possible way of regulating this top-down control, but also potentially, therefore, you can use oxytocin to do the same thing. Just to quickly remind those of you who are perhaps not so aware of the oxytocin field, it was discovered by Sir Henry Dale some over 100 years ago. And of course, we know it best for its smooth muscle effects in terms of uterine contractions and milk ejection reflex. But starting really from the 1980s with animal models, we discovered that it was released within the brain. It has independent pathways within the brain, as well as, I already mentioned, this paracrine cerebroventricular distribution system. In terms of oxytocin receptors, this is human postmortem tissue. This just shows you they're very widely distributed in the brain, and I've indicated which systems they involve. And you can see executive attention, salience, emotion, reward, social and sensory systems, pretty much all of the social brain. We give peptides like oxytocin and insulin, vasopressin via intranasal infusion, and it tends to take around 45 to 75 minutes for the peptides to actually reach their maximum concentration within the CSF, but it's important to emphasize that you also get increases in the vascular system. This is a regional cerebral blood flow study by Palayelis, just to show you the time course of the changes you get within all of these areas of the brain containing oxytocin receptors, and around 39 to 51 minutes after the nasal infusion. So oxytocin, we know, is released in humans and animals: sex, birth, suckling, tactile stimulation, social interactions. And either genetic association or intranasal infusion studies have shown that it's involved in a wide range of particular behaviors. But I want to focus here on what the relationship is, if any, between resting state and task effects of oxytocin. And to do this, it's a study by one of my students, Joy, who's also got a poster here. We looked at the effects of oxytocin both on resting state and on responses to fear faces on 2 occasions, on the first day and also 5 days later. And one group, a control group, gets placebo nasal spray every day, once a day. Another group gets oxytocin on alternate days, placebo the other days. And finally, a group gets oxytocin every day. You might wonder why we're interested in this. Well, it's because so far there haven't been any studies looking at the possibility that dose frequency may have an influence on responses in the brain. And it's well known that oxytocin receptors can desensitize, at least in certain parts of the brain, notably the amygdala. This shows you very nicely that for the responses to fear faces, you get a reduction in the amygdala response, both on the first day in the alternate oxytocin OT3 group and the everyday oxytocin group. But interestingly, in the group given every day, you don't get a response, significant response on the 5th day, indicating desensitization, whereas in the group given every other day, you still do. So dose frequency may be important, at least in terms of the amygdala reduced response to fear faces. There's also a corresponding behavioral effect in that the emotional arousal and intensity ratings given by subjects subsequently are also reduced by oxytocin, and again, more effectively in the group given the infrequent oxytocin doses. And this is also shown, at least on day 5, by an improved association between the changes and the intensity. Now we turn to the resting state. Very impressive effects, as you can see, quite a large increase, a doubling of the strength of the resting state functional connectivity with oxytocin. And you'll notice immediately that here we don't see a desensitization effect. Doesn't matter whether you give the doses every other day or every day, you get a nice increase in resting state functional connectivity. And if we look at another set of associations, which is here, these are healthy subjects, and we're looking at associations with trait anxiety and also the autism spectrum quotient. Here we find that only with the resting state changes do we find some kind of association between the oxytocin effect and the scores on these 2 traits, not with the response of the amygdala to fear faces. On the other hand, only in the amygdala response reduction to fear faces can we find genetic associations. In fact, with 2 different SMPs of oxytocin receptor, RS53576, as well as 2254298, A carriers show the oxytocin effects, whereas non-carriers don't, which has a very important observation going forward if we're going to be using oxytocin in a therapeutic context. However, at least with these SNPs, we find no association with the resting state. And finally, if we actually use PPI to look at the same functional connectivity under task conditions between the same location of the medial prefrontal cortex and the amygdala, and we look at the correlation between the functional connectivity in the resting state and in the task situation, as you can see, there is absolutely no relationship at all. So for this first part of the experiment, we can conclude that, yes, single doses of oxytocin do robustly decrease amygdala responses to fear faces, and they also robustly increase resting state functional connectivity between MPFC and amygdala. Repeated doses show reduced amygdala responses are subject to desensitization. The oxytocin effects on amygdala responses to fear faces, but not resting state, are associated with specific alleles of the oxytocin receptor gene, or at least they're going to be possibly associated with different ones if we looked at all of them. The oxytocin effects on resting state functional connectivity, but not amygdala responses to fear, are associated with trait anxiety and autism. And finally, there's no correlation, at least with the different ways of measuring functional connectivity, between the functional connectivity and MPFC and amygdala in the two conditions. But using the same data, what about task versus rest comparisons at a network level? And here, with Jiangxi in my lab, we've used a change point model analysis, which I don't have time to go into, but it allows us to look at directed connectivity in both resting state and task, slightly different approaches, of course, to identify the state changes in each case. When we're using this approach, there's no effect on state change frequencies. But in terms of resting state connectivity patterns, if we look at the sort of summary of the changes during PLC, the unique patterns, this is in 80% of subjects, are pretty much all within regions that we analyzed. Whereas when you give oxytocin, there is this nice top-down from medial prefrontal to amygdala enhanced connection, and also, in fact, between brain reward areas in the amygdala and between the two key nodes of the salience network, the anterior cingulate and also the anterior insula. If we look on the same situation on the task, it's similar in many ways. Again, you get the connections within regions primarily showing up with the PLC, but also some within the salience network. But when you give oxytocin, you get in connectivity between the ventral medial prefrontal and the anterior cingulate. Interestingly, the anterior insular connectivity with the anterior cingulate is reversed. So again, we see no great overlap between the effects of oxytocin on rest and task on networks. So overall, we can conclude so far, and we still have obviously a long way to go, that the directionality of the oxytocin effect on resting state between MPFC and amygdala is as predicted top-down, which is very important for us to establish. It hasn't so far been established as being top-down. Patterns of functional connectivity produced by oxytocin during rest and task both involve interregional interactions. However, the specific functional connectivities that are influenced are either different or may even have different directionality. And finally, of course, we need more task and resting comparisons, and maybe particularly we'll see a closer relationship between task and rest in perturbed systems in patient populations. But in any event, even though I've tried to show you that they are very different in terms of what oxytocin is at least affecting in the rest and task situations, clearly it makes sense to measure both, because the combination of the two may give you even better discrimination accuracy. So rather than actually saying we should either measure one or the other, I think this is a good argument for why we should be measuring both. Finally, it just remains to thank my group, NuScan, at UESTC in China, both in resting and tasking situations. Thank you very much indeed.